So this is part 3 of tricky concepts related to renal physiology, the kidneys. We're going to talk about the countercurrent multiplier in the loop of Henle, its role overall, and lastly we're going to talk about the role of the hormone antidiuretic hormone. So first let's talk about the countercurrent multiplier in the kidney. That's going to take us to the loop of Henle. Let me draw our nephron on the board again. All right, there's our schematic nephron. Of course, we know the nephrons are responsible for producing a urine, but the urine is the final product. Initially, what flows through the nephron is referred to as a filtrate. And of course, this solution is derived from the plasma. There's our blood supply to the nephron. The afferent arteriole feeds the glomerular capillaries, and the process of filtration, this pressure-dependent process, produces the initial filtrate that enters Bowman's capsule. Now let's talk in terms of solute concentration in that initial filtrate. This initial filtrate, again, is derived from plasma. And in the human body, plasma has an approximate solute concentration of 300 milliosmolar. The milliosmolar being the concentration, or milliosmoles per liter, milliosmolar. The number is it's kind of a funny number, and they're funny units. But again, it reflects a solute concentration. So 300 milliosmolar is the plasma solute concentration, and the main solute that contributes to that overall concentration is the sodium ion. If that number gets larger, if the plasma osmolality goes to 305, that means the plasma becomes more concentrated. There's more solute per liter. Similarly, if that number gets reduced, if it goes to 295, that means the plasma has become more dilute with regard to solute concentration. All right, so that's the plasma solute concentration. Plasma is filtered by those glomerular capillaries, and the initial filtrate that develops in Bowman's capsule has the same overall solute concentration. So I'll write 300 right there in Bowman's capsule going into the proximal tubule. Now you'll recall about 70% of everything that enters the proximal tubule gets reabsorbed. It's a very active area of the nephron. 30% proceeds past the proximal tubule into the loop of Henle. Now 30% of everything getting reabsorbed, ions, glucose, water, etc., that means that the remaining filtrate that makes its way to the loop of Henle has a osmotic concentration of 300 milliosmolar. It hasn't changed yet. Now, the descending limb of the loop of Henle and the ascending limb of the loop of Henle differ with regard to their water and sodium permeabilities. The ascending limb of the loop of Henle has sodium pumps. It actively pumps sodium ion out of the filtrate. Chloride, by the way, will just follow passively this movement of sodium. The descending limb of the loop of Henle is permeable to water. Water will move out of the descending limb because sodium ion is being pumped out of the ascending limb. That is, the sodium ion concentration will develop in this interstitial space here between the limbs of the loops and all over, and water will move towards this area of higher solute concentration. So as the filtrate moves down the descending limb, where water is moving by osmosis out to the interstitium, that filtrate becomes more concentrated. Water is leaving. Until it reaches a peak concentration at the bottom of the loop. That's the end of the area for water permeability. And the filtrate in the loop of Henle now may reach as high as 1200 milliosmolar, especially in those very long loops. On the way up the ascending limb, sodium ions are pumped out of the filtrate. Selectively, sodium ions are pumped out. This will dilute the filtrate. And this is a funny concept, that sodium ions are moved out of the filtrate. But moving those sodium ions out leaves a more dilute filtrate behind in the nephron. So again, as the filtrate moves its way up the ascending limb, it becomes progressively more dilute. Until that filtrate reaches the top of the ascending limb, just prior to entering the distal tubule. At this point, the filtrate is actually more dilute than the initial filtrate that entered the nephron, which, by the way, you remember, was the same osmotic concentration as the plasma. So here we've produced a filtrate entering the distal tubule that is hypoosmotic. It has a lower total solute concentration than the plasma. This filtrate then has a lower total solute concentration than the whole rest of the body. So let's stop it right there and think for a second. What if this filtrate now left the distal tubule unchanged, entered the collecting duct unchanged, and made its way to the bladder as urine? What effectively have we done to this individual who started with a total solute concentration in their body of 300 and is producing a urine of 100? This individual is effectively losing more water. They're producing a urine that's less solute concentrated than the rest of their body. We are concentrating the whole individual by producing a urine that's less concentrated than they are. So if that filtrate leaves the distal tubule and the collecting duct unchanged, if we produce a very dilute urine relative to the rest of the body, we are concentrating this individual. All right, now imagine if we could modify that filtrate that goes from the ascending limb loop of Henle into the distal tubule and collecting duct. What if we could absorb some more water out of the distal tubule and the collecting duct and modify that filtrate, if we could concentrate it some more? We can, and there's a hormone that plays a role with this, and more on that in just a second. But the ability to modify that filtrate that leaves the loop of Henle into the distal tubule and collecting duct all depends on the interstitial osmotic gradient that the loops of Henle have developed. Because as the loops of Henle develop the gradient within the nephron, a similar gradient develops out in the interstitium, because sodium is being continuously pumped out of the ascending limbs. Now remember, there's 1 to 2 million of these nephrons. And so what happens is that an osmotic gradient is developed through the interstitial region of the kidney, with it being isosmotic to the body, the same solute concentration, all the way down to a concentration that can be as large as the loops of Henle can produce. This is the gradient that can allow for the alteration, the modulation of this final filtrate that enters the distal tubule and collecting duct. Water always moves by osmosis. This is the gradient that will be the driving force for osmosis, allowing water to be reabsorbed in the distal tubule and the collecting duct if a hormone is present.